We as humans are considered to be a single organism. When you really think about it, however, our bodies are really made up of thousands upon thousands of individual tiny cells that help to perform all tasks necessary in order to survive. Nearly all of the cells in our bodies are constantly dividing in order to do things such as enable us to grow and replace worn out or damaged cells by means of what is known as mitosis. Mitosis, however, is part of a larger cell cycle, which can be split into two broad phases. The first of these phases is interphase, as shown in the diagram. Interphase can further be divided into the G1 phase, during which the cell adds to its supply of proteins, creates more organelles in its cytoplasm, and grows in size, the S phase, during which DNA is synthesized, and the G2 phase, during which the cell finishes growing and completes its preparations for cell division. During interphase, two centrosomes exist in the cell's cytoplasm, and duplicated chromosomes appear as loosely packed chromatin. Two or more nucleoli may be present in the nucleus of the cell, which is evidence that the cell is continuously producing proteins. The first stage of mitosis is prophase. During this phase, numerous changes occur in the cytoplasm and nucleus of a cell. Within the cytoplasm, the formation of a mitotic spindle begins when microtubules start to grow rapidly out from the two centrosomes. These centrosomes begin to move away from each other towards opposite poles or ends of the cell. Inside of the cell's nucleus, chromatin fibers coil and fold more tightly and form easily recognizable chromosomes. These now duplicated chromosomes appear as identical sister chromatids joined together at the centromere found at the narrow waist. During prophase, the nucleoli also disappear. The second phase of mitosis is prometaphase. During prometaphase, microtubules that have emerged from the centrosomes at the ends of the spindle reach the cell's chromosomes, which are now highly condensed. They then attach to a protein structure found at the centromere of the chromosome, called the kinetochore, shown as blue dots in the movie. This action sends the chromosomes into motion, and soon forces from protein motors associated with the microtubules of the spindle move the chromosomes toward the center of the cell. During prometaphase, the nuclear envelope also breaks down into pieces and eventually disappears. The third phase of mitosis is metaphase. During this stage, the formation of the mitotic spindle is complete, and the centrosomes are at complete opposite poles of the cell. The cell's chromosomes meet at the imaginary metaphase plate, which is equidistant from both poles of the cell. Here, their centrosomes align, and the kinetochores of each of the two sister chromatids face opposite ends of the cell. Microtubules from the mitotic spindle attach to different chromatids, while microtubules from the opposite end of the spindle attach to those chromatids' sister chromatids. The fourth phase of mitosis is anaphase. The beginning of this stage of mitosis is marked by the two centromeres of each of the chromosomes coming apart and thus separating the sister chromatids. Once they are separated, the sister chromatids are now referred to as full-fledged daughter chromosomes. Motor proteins in the kinetochores, which are powered by ATP, now slowly guide each daughter chromosome along the microtubule it is attached to, centromere first, toward opposite poles of the cell. While this is happening, the microtubules of the mitotic spindle attached to the chromosomes shorten, while those not attached lengthen. This forces the poles of the cell to move farther apart, causing the cell to elongate. Anaphase then ends when the collections of chromosomes reach their separate ends of the cell. The fifth and final stage of mitosis is telophase. During telophase, the cell continues to elongate, while daughter nuclei appear at opposite ends of the cell. Nuclear envelopes then form around the chromosomes. While this is happening, chromatin fibers in each of the chromosomes uncoil, and a nucleolus appears within each of the nuclear envelopes. At the end of this stage, the mitotic spindle disappears entirely, and mitosis is complete. The second part of the mitotic stage of the cell cycle is cytokinesis. This process involves the division of the cytoplasm and usually coincides with telophase. During cytokinesis, the two daughter cells completely separate as the cytoplasm is split into two equal parts. This can happen differently in plant and animal cells. In animal cells, a cleavage furrow, which is a shallow groove in the cell's surface, forms a ring of microfilaments made of actin and contracts, pinching the parent cell in two. In plant cells, however, this same process cannot occur because of the cell wall. Within plant cells, vesicles containing cell wall material form in between the two new daughter nuclei. These vesicles fuse and form a cell plate. This cell plate then grows outward and eventually fuses with the plasma membrane. Either way cytokinesis occurs, the result is two daughter cells that are genetically identical. Now that we know a little bit more about each stage of mitosis, let's see it all put together.